Welcome back to the studio. Thank you so much for joining us in the studio. We've got Judith, Stephen and Pamela who've just been on stage. Now, you guys have just done a lightning talk. What was it? You had 4 minutes, 5 minutes?
Yeah, 4 minutes, one question.
4 minutes, one question. I mean, that is not a long time. How do you prepare for a lightning talk?
I tried to rehearse and then forgot everything just before. That's pretty much how it went.
I take the complete opposite. I didn't rehearse, so I like to just... I know what my slides need to say. I just rock up and hope for the best.
You know, adrenaline is a powerful force when you're standing up on that stage in front of a 25-metre screen in front of 600 people. That's fantastic. Well done. Your talks were fantastic. We will come back to you in a minute. But Judith, thank you so much from coming from Great Ormond Street Children's Hospital, where you do so much fantastic work there, to share your experiences with the audience. And I would really love to understand the sort of human story behind the science that you have talked about on stage today. So you were saying earlier that you really connect very deeply with the patients that you are involved in their care pathway. Can you tell us a story of a patient and a family who's been through this journey and how they've benefited from this rapid metagenomic approach?
Yes, I mean, well, you know, we started doing this about 10 years ago because we have quite a few patients who have a sort of encephalitis, a brain inflammation, and everything is negative. And then you're left with the question, is it an infection, is it inflammation, or is it the drugs that they're on? And often what used to happen was you would just say, okay, we don't know what to do, we're just going to try one of those. And if it was the wrong one, then that led to, you know, sometimes the patient dying. So actually the very first time we did this was on a child who had an encephalitis. They had an underlying immunodeficiency. They had what we call a primary immunodeficiency, and they'd had a bone marrow transplant, and they were on quite toxic drugs, and literally we did not really know what was going on. And I was doing RNA sequencing for my research, and I just said, we should just do it. We should, you know, what do we lose? This child is going to have a brain biopsy anyway because we don't know what's going on. Why don't we just put some of it through? And we did. And we got a pathogen the very first time, and it was a pathogen that had not really been found before to cause brain infections. We found that same pathogen in another child, and now it's been found in lots and lots of people, adults and children, who are immunocompromised. We found that same pathogen in another child about 5 years later, under exactly the same circumstances, but the child was not as sick as the one we'd first done it on. And what we were able then to do was to say, right, this is a virus that theoretically could respond to some drugs that we have. Never been tried before, but we should try them because this child is a one-way street if we don't actually try this. And we were able to give this child drugs, and it stabilized her, and then her immune system kicked in and she was able to clear the virus. So what it has done means that we are now able to offer hope to some of these very sick kids. When we find something, we can actually also use experimental drugs that might also help prolong their life as well.
I mean, that's a fantastic story, and thank you for sharing it with us. And I think it makes us all want to come in and do the work and support each other in reaching those outcomes. And how do you think, I mean, you talked very articulately on stage around how you're thinking about scaling this into what was clearly a sort of an urgent and single case into something that could be reaching all children and all patients. What are the challenges with scaling, but also the opportunities?
Well, the respiratory metagenomics has given us a lot of insight into how to scale things, and that's been a really good eye-opener. And what you need is a method that is robust and transferable. And actually, we were very fortunate that we stood this up very quickly, so we were able to prove to Guy's and St Thomas's that the method they developed was really robust and transferable. We are now working on doing the same for the sterile sites metagenomics, and that's where the nanopore actually has come in, because we think that might be more scalable and transferable and easier to draw up a standard methodology that can then be rolled out. And so that's what we're doing this year. We're actually making it into a standard methodology. We're testing it against our existing pipeline, and then once we know that it's working well, we will then roll it out to other centres across the UK.
it's just wonderful to see the implementation, this sort of being realised. And Stephen, I mean, you've been talking about ultra long in leukemia. What's the dream of taking? Where would you like your work to end up in the clinical system?
I mean, the dream is having it in a clinical setting. First of all, I think what we're doing is kind of the foundation work to show that it does work, and it works very well. We are aware of things like sample scarcity, so it's not always easy to get enough DNA, but there are other techniques that we're also developing to combat that. So
Yeah.
actually we can get around that fact using slightly modified protocols and that sort of thing. But I think what it's shown us is that actually everything we've looked for so far, we've been able to detect. There is also cohorts of patients that still they get a leukemia diagnosis, but they're not subtype defined, and they're treated with high intensity treatments because we don't know what else to do, basically. And actually, this opens the avenue to start looking into those cases as well, where at the moment they don't receive a proper diagnosis, but actually I fully believe this technology will unlock that diagnosis as well. And I'd love to see, I mean, I guess the dream is, every doctor's office around the country would have one of these devices. That would be wonderful. But until we're at that point, I still think, we're talking about quick turnaround times, full diagnosis, and full characterization of people's disease. And I think it unlocks the door for further therapies, precision medicine, and targeted therapies. So that's the dream. Yeah.
There are very few scenarios in the clinical world where having an answer faster is not beneficial, whether it's for the emotional journey that the families are going on, whether it's for the clinical decision making. That seems to be a very common
Absolutely.
theme.
And I think for some of these patients, they will require so many tests to then find out, actually, we still can't give you a definitive diagnosis. And I think, so from the patient's point of view and the family's point of view, expediting that process, but also being able to give them an answer at the end is really crucial
Fantastic.
for them. Yeah.
It's also really interesting to see the theme that this has come up again and again in the last 24 hours around doing the testing so that treatment pathways could be made less onerous in certain situations, whether it's it's actually immunology rather than an infection, or whether it maybe is a form of leukemia that the more robust treatments aren't needed for. This is an increasing theme, and I think it's
absolutely.
really interesting.
Treatment de-escalation,
Yes.
I think, is crucial because we're using the same cancer therapies we used 30 years ago, and we all know they're very, very harsh. They're very, very damaging to your body.
Yes, yeah.
And anything we can do to lessen that, I think, is a huge benefit, and this really helps with that.
With potential economic impacts onto the system as well. I mean, if we're thinking about health economics. Sorry, Judith, you were nodding.
The precision medicine side of it,
Yeah.
I think this is what's really important, actually being able to give a specific diagnosis to each patient, so that you can actually tailor treatment rather than just having the sort of blunderbuss approach to everybody.
Well, talking of precision treatment, pharmacogenomics, obviously, Pamela, thank you so much for your talk on that topic. So in your world, you're working on improving pharmacogenomic methods. How do you think that could benefit the precision medicine approach? And particularly in Singapore where you work and in Asia, are there different approaches in the healthcare systems there that pharmacogenomics might be specifically relevant for?
Oh, okay. So basically, I think pharmacogenomics is definitely something that will benefit the patient if it's done preemptively, right? Because we know, I think it's the message that we've talked about as well. It's like what people may have a diagnosis, and then they'll be given a slew of drugs based on the indication that they've been prescribed. So, however, not all people have the same response to that specific drug, and the idea is that rather than going back to the doctor again and again and trying to adjust the dosage slowly to suit your needs or what is right for you, have a test before you have to go through that, and make an informed decision. Like, have the doctor as well, make an informed decision about how they can best adapt the treatment for you as a patient.
Absolutely. Judith, are you using pharmacogenomics approaches routinely at Great Ormond Street?
Yes, I think in some circumstances,
Yeah.
absolutely. But I think it's not routine, but it's in particular indications it's used. But I think we would like it to be routine. You know, if you've got that as a part of the result that came out on everybody, whether they had particular susceptibility or whatever it is to certain side effects, then that would be an amazing jump
So
forward.
how many years are we away from a routine telomere to telomere lifetime genome in your electronic health record that services your pharmacogenomic drug planning? 1 year, 5 years, 20 years?
It's also a question of finance, isn't it? I mean, who's going to pay for that? So
Yes.
that's always what comes up.
Yes.
And so, yeah, I mean, I mean, technically we can do it.
Yeah.
You know, we could do it now, but it's a question of whether who's going to pay, and when that's going to happen.
Well, as we've learned from the last 24 hours, the thing that's going to propel that is champions and people pushing the cause and telling wonderful stories about why we should be doing this kind of innovation and bringing it to the healthcare system. So thank you so much for sharing your science, but also sharing your stories.
Thank you.
Hopefully the online audiences have been paying attention and been enjoying it. Thank you so much.
Thank you.
So unfortunately, we've run out of time for our short chat, but I do have some other messages. So coming up next, you can connect with other delegates online in the networking room. You can explore the poster hall. You can find all the inspiring delegate posters there. And of course, don't forget to vote for your favourite. In the auditorium, in about 5 minutes, you can watch yesterday's amazing on-site session in precision genomics. Alternatively, you can also see 2 fantastic presentations on the use of nanopore sequencing in neuroscience. Then at just 11:05 BST, we'll move into our second round of breakout talks. So make sure you check the agenda for all the details there. And don't forget to carry on posting on LinkedIn, Twitter, Blue Sky, using #NanoporeConf for your posts. Thanks very much, and we'll see you soon.